Hi, everyone. I'm happy to introduce our speaker today. Andrew Durso just joined the faculty at FGCU in January of this year. And we were planning to have him come visit Archbold a couple months ago, just to let him see our research site, see about any potential research opportunities and give a seminar. But we ended up having to do a rain check because of COVID. However, he kindly agreed to go ahead and do his seminar virtually for the benefit of a bigger group of people. So Andrew is a reptile biologist. He uses a combination of field and lab approaches, including stable isotopes, to study physiological trade-offs and the population community ecology of reptiles. He also has strong interest in human-wildlife interactions and citizen science. And he's recently been delving into global public health related topics and applications of machine learning and artificial intelligence. So Andrew holds a master's from Eastern Illinois University and a PhD from Utah State University. And he recently returned to the US from 2 years in Europe, where he completed a postdoc at University of Geneva in Switzerland. So something that's really impressive about Andrew is he's gotten a lot of recognitions for his college teaching abilities, and he does amazing outreach with the public to improve knowledge and really rectify misperceptions about snakes and other reptiles. And as you know, this is a group of organisms that often needs all the good PR they can get. So with that, I'm going to turn this over to Andrew, and he's going to be talking about semi-aquatic snake communities. Thanks, Andrew.
Thank you so much, Betsy, and thank you, Laura, for introducing me and hosting me today. I certainly would have preferred to come to Archbold in person back in March. Hopefully I'll make it out there eventually. It's only a couple hours away from Fort Myers, where I am now. And yeah, today I'm going to talk to you about some of my oldest work, actually. So my undergraduate work from the University of Georgia, Savannah River Ecology Laboratory, on the ecology of semi-aquatic snake communities, with an eye towards hopefully getting some of this work restarted and trying to address similar questions in Florida. I grew up in the Southeast, and I haven't lived here since I left college, and I'm really excited to be back because we have a ton of cool herps and a ton of awesome snakes like this mud snake that we actually still know very little about, and I'm really motivated by trying to learn more about the ecology of some of these beautiful and secretive organisms. So let's see. So this is me in 2004 in high school. I grew up, as I mentioned, in the Southeast in North Carolina, catching snakes. I was involved from a young age at the North Carolina Museum of Natural Sciences and the North Carolina Herp Society. And that's where I first heard about the University of Georgia. And I decided that I would really like to work there someday, particularly at the Savannah River Ecology Laboratory, which has a long history, which I'll go into in a moment. But first, let me just mention, Betsy sort of covered this, but I have spent 4 years at the University of Georgia doing my bachelor's degree in ecology, and then I moved through Eastern Illinois University, where I did my master's, and Utah State University, where I did my PhD, to Europe, where my wife did her PhD, and so I was based briefly at the Max Planck Institute for Biogeochemistry in Jena, Germany, where she was a PhD student before doing my postdoc at the University of Geneva in Switzerland, and returned to the US this January to start teaching at Florida Gulf Coast. But throughout that entire time, I have been sort of thinking about three main questions, and they're really basic questions. So they are: which snakes are where, how many are there, and what do they eat? And these sound like such simple questions, and you're probably thinking, gosh, if snake biologists don't know the answers to these questions by now, you know, what are they doing with their time, right? And the answer is that we do have some answers to some of these questions, and I'm going to try to provide you with a little bit more detail today. But these are challenging questions to answer because snakes are very secretive, they're very difficult to find, and so it's hard to do even very basic ecology on them. So I'm going to suggest some approaches that may help. And I did all of this work at the Savannah River site in South Carolina. So the Savannah River site is a US Department of Energy property. It was established in 1950 and operated by the DuPont company until the late 1980s, after which it was taken over by the Department of Energy. It's the home to 5 nuclear reactors that have been used to produce tritium and plutonium for nuclear weapons. Until the end of the Cold War in 1991, the reactors are now used for waste storage. And because of the dangerous nature of this work, the Savannah River site was really established as a buffer zone to prevent nuclear radiation from spreading into communities inhabited by humans. So it's very, very large. It's 310 square miles, or almost 200,000 acres. It actually makes up 1% of the state of South Carolina. And access is restricted only to people with badges. You know, we don't need no stinking badges, but you do need a stinking badge if you want to go on the Savannah River site. And this makes it a really good place to do ecological work because there are a lot of undisturbed habitats there, or relatively undisturbed. I will say that you may notice the name of this town just to the north of the Savannah River site is New Ellington, and that's because many people were relocated to establish this site. So about 1,500 families were relocated from small towns in the Savannah River site in the 1950s. And Ellington was the largest of those towns. It had a population of about 760. And most of these people were African Americans. They were poor tenant farmers. And this book sort of details one person's experience through that unexpected exodus from Ellington to New Ellington. And if you look at the map of the surrounding area, you see a lot of other new towns and new cemeteries. They relocated over 150 cemeteries to create this site. So, you know, very interesting culturally, and has left a major mark on the surrounding area. And you can really see the Savannah River site well, also from space. So I don't know if you can see my pointer here. You can see the Savannah River, which is the state border between South Carolina, on the north, and Georgia on the south, running from the upper left corner of the screen down to the bottom center. You can see Augusta, Georgia, and across the river from it, Aiken, South Carolina in the upper left. And you can see the Savannah River site is this big round thing together with this tail, which is lower Three Runs Creek. And you can see some of the large ponds that were created for nuclear water that was used to cool the rods for the nuclear reactors, as well as some of the infrastructure and things over here. So yeah, so the Savannah River Ecology Lab is an institution that was established just a year after the founding of the Savannah River site. It was established in 1951 by Eugene Odom, a professor of ecology at the University of Georgia, who later became known as the father of modern ecology. He wrote the first ecology textbook and did some classic experiments, including experiments with radionuclide tracers that kind of propelled the shift in ecology from looking more at structure and sort of describing what species were found where, to looking more at the function of ecosystems and how energy and matter flowed from one place to the next, and how organisms, plants and animals and other organisms interacted with one another. Odom's initial budget was $10,000. By the time the lab became permanent in the 1960s, the annual budget was over $100,000. It has become part of the long-term ecological research network and was the first member of the National Environmental Research Park. The goals of which are really to provide baseline data for environmental change in North America, and specifically at the Savannah River site to independently evaluate the impact of Savannah River site operations, so nuclear operations, on the environment and environmental health, and by extension, human health. So this is really important, and I think very foresightful that the Department of Energy saw fit to fund this. By the time I was there in 2006, the budget had already sort of peaked. I think I read it peaked at something like $12 million a year in 2002, and then has slowly declined since that time. But the reason that I wanted to go there is that SRL has been a really important center for herpetological research since 1967, when Whit Gibbons joined the laboratory as a postdoc, fresh out of his PhD at the University of Michigan. Since that time, researchers have captured, marked, and released more than 1 million individuals of over 100 species of reptiles and amphibians on the Savannah River site. And they've also done a lot of other good things, a lot of education and outreach in the communities. I think every school child in rural South Carolina probably was visited by Whit or someone working under him at some point. And they also funded this research experience for undergraduates through the National Science Foundation, which is what brought me to SREL in the summer of 2006. Where I overlapped briefly with Betsy, although I don't think we didn't spend a lot of time together, certainly not in the field at that time, and we've gotten to know each other better since then. So that's kind of my Archbold connection. Okay, so the Savannah River site is very diverse. The natural communities range from upland sandhills and hardwood and pine forests that are very well drained to bottomland or swamp forests that are periodically flooded. There's over 55 reptile species, over 40 amphibian species, and the longest term study of any amphibian community is still taking place, as far as I know, on the Savannah River site at Rainbow Bay. And so these are data published in the 90s from that study, and you can see that they show sort of over time the cumulative number of species and individuals reported from Rainbow Bay. And I want to draw your attention to something that happened in 1991-92, which is that the species accumulation curve for reptiles really didn't finish being filled out until they'd been surveying there for almost 20 years and had captured over 300,000 individuals. And so at that point, they were still adding new species, in particular a new species of snake, the southeastern crown snake, Tantilla coronata, which is a relatively cryptic, certainly, and in some places, many places, really a rare species. And that's just amazing to me. I mean, how are we to say with confidence which snake species occur where when you can catch almost 500,000 individual reptiles in a really intensive survey and still be adding new species at that point? I really, like to point to this because it is just such a good illustration of how difficult to sample some of these animals are. Okay, so Rainbow Bay is a very unique type of ecosystem called a Carolina Bay. Carolina Bays are these depressional wetlands that share a number of unique features. You can see 3 or 4 of them in the satellite photo on the right. They all have this kind of northwest-southeast orientation. They're all sort of vaguely egg-shaped with a limestone rim at the northwestern edge and a sand rim at the southeastern edge. There's about somewhere estimated between 500,000 and 2.5 million Carolina Bays, mostly in the Carolinas, which is why they're called Carolina Bays, but some in Virginia and also some in Georgia. Interestingly, they're not, the word bay actually comes from observations of bay trees growing along their margins and not because they're wetlands, even though a bay also means a saltwater wetland. And it's a little bit mysterious where Carolina Bays came from. Some people think that they may have been formed by a meteor shower. I was reading about it this morning and came to find out that that seems like it's mostly been debunked now, and it seems more likely that they were formed during the receding shoreline that exposed the portion of the continental shelf that we now call the coastal plain, and they were formed by kind of the action of wind and water during this time. But maybe the jury's still out a little bit. I don't know. I'm not a geomorphologist, so if you want to know, you should ask one. What I do know about Carolina Bays is that they're really threatened. 80% of them have been drained or converted and used for agriculture or developed in some other way. And they're also really, really cool ecosystems because they support a ton of rare plant and animal species, including a lot of carnivorous plants and a lot of amphibians and reptiles, really good habitat for migratory birds. And they're really, really good replicates to use in sort of large-scale ecological field studies. Even though they're all similar, you can see that they vary in size and they do vary in other things as well. Some are permanent, others are ephemeral, meaning that they dry up pretty reliably, maybe every year or more often than that. So each is also unique, and you can kind of build a gradient, which I'll show you is exactly where we went in our studies of aquatic snakes that inhabit these Carolina Bays. So if you're in the Georgia or the Carolinas or Virginia, maybe go try and check out a Carolina Bay near you. They're super cool. So these are data on hydroperiod from 10 Carolina Bays on the Savannah River site. You can see that it fluctuates a lot from year to year, and some of them fluctuate all the way to zero, so they dry out completely, others less so. There's one in there that is probably maybe fed by a spring and it fluctuates less than the others. So just give you some idea of their depth and how it can change from year to year and also across decades. They're not very deep. It'd be hard to go into the water over your head in any given Carolina Bay, maybe in a really wet year. And one of the most famous, arguably the most famous Carolina Bay, I suppose, is one that's on the Savannah River side called Ellington Bay. So Ellington Bay, depending on how full it is, it varies between about 5 and 10 hectares. It's normally less than a meter deep, maximum maybe 2 meters in the very center in a wet year. And it's also interesting because, like many wetlands that are important to amphibians and reptiles, it's relatively isolated. So it's at least 3 kilometers from the nearest permanent wetland. Mostly emergent vegetation, grasses and water lilies, very few trees growing within the footprint of the bay itself. Eugene Odom conducted a number of his famous old field experiments in the former fields that were around the bay. You can see from the satellite photo now that most of those fields are gone. They are so old that they have succeeded now to forest, but some of his original plots and some of the bits and pieces of his experiments can still be seen there. And it has been sort of continuously, more or less continuously, maybe not quite so much as Rainbow Bay, monitored for amphibians and reptiles since 1968. So there's 23 amphibian and 20 reptile species in Ellington Bay. Importantly, there are no fish, except for Gambusia, the live-bearing mosquito fish, in Ellington Bay. And I'm going to return to the importance of fishless wetlands in a minute, but if you know anything about amphibians, you're probably already familiar with the idea that fishless wetlands are really important habitat for amphibians, and that's also true for predators of amphibians like aquatic snakes. The first person to really work on aquatic snakes in Ellington Bay was Rich Siegel during his studies for his PhD in the 1980s. And since that time, Whit Gibbons and others have done a lot of really good work on aquatic snakes in Ellington Bay. And at the time that I joined the lab in 2006, one of the first conversations that I had was with a PhD student, J.D. Wilson, who told me that he's really had been curious about a lot of the other Carolina bays on the Savannah River site, because most people found it easy and convenient to work at Ellington Bay. It's right off the road. There's a lot of historical data, and there wasn't a lot of motivation to develop new field sites. And so he wanted to kind of explore some of these others. So we picked out an additional several Carolina bays that were similar to Ellington Bay that dried out. And we also picked a number of more permanent wetlands that didn't dry out in the summertime. And so some of these are not maybe strictly Carolina bays, but a lot of them are wetlands that are very similar in size and structure and vegetation community, and also snake community. Importantly, there's also kind of a gradient here. So many of these permanent ones had fishes, not all of them, depends on how isolated they are. None of the ephemeral ones do, for obvious reasons. And these were kind of our replicates in our experiments that we conducted on aquatic snakes. And they ranged in size from 0.03 of a hectare all the way up to 78 hectares. So the size is to scale here, but the distance is not. Here they are on an actual map, so you can get a better sense of how they are spread out across the environment. The yellow ones are the ephemeral ones without fishes, and the green ones have fishes, and some photographs showing a more permanent wetland on top and a less permanent one on the bottom. So the way that we would collect snakes from these wetlands is we would use these aquatic plastic minnow traps that have a funnel at either end. It's kind of hard to visualize how these work unless you're holding one in your hand. But basically, they have two halves that snap together. There's a funnel on either end that points inward. And so a snake can get in, but it's much harder for it to get out. They can get out, but most of the time they don't, in particular because these traps also have kind of the advantage of being self-baiting, so lots of other organisms like to go into them, fishes, if they're present, amphibians, frogs, salamanders, things that snakes like to eat. And so they'll go in there, and sometimes the snakes will go in there to eat them, other times they'll just go in there in their course of their everyday movements within the wetland, foraging or looking for mates or whatever they do. So these are a really good method for detecting snakes. They're relatively low effort. They're highly, highly repeatable because all the traps are the same. Observer bias is really low. It's pretty easy to check a trap, even if you've never checked one before. And yeah, like I mentioned, they're self-baiting. So quite a good method. And so you can see this kind of trap line in the water on the left here. Usually we were setting 30 traps at a time, sometimes more, to detect and mark and recapture aquatic snakes. And you can see one in the bottom right there, kind of tail sticking out there. That's actually a cottonmouth that's entered the trap to eat a water snake that's trapped within the trap and gotten stuck. So before I show you the data, I'll just introduce kind of the cast of characters. So these are the 7 species of aquatic snakes that we focused on in our studies. The most common species is the banded water snake, Nereida fasciata, which made up 60% of captures, so by far the most common species. Second most common species, is this cute little guy, the black swamp snake, Liodides pygia. When I was working on it, it was called semi-natrix pygia, so I'll probably screw up and call it semi-natrix at least once or twice during this presentation, and you might see it labeled as such on certain graphs. These are kind of rare at the landscape level, but they're pretty common in certain wetlands on the Savannah River site, so they made up about 20% of all captures. And then there are 5 less common species, so Florida green water snakes, which are kind of fish specialists, we think, that live mostly in permanent water bodies. They made up 3% of our captures. Another species that used to be in a different genus, Liodides rigida, used to be Regina rigida, the glossy crayfish snake, so a crayfish specialist, making up 1% of captures. The large and beautiful mud snake, Varantia abacura, a very interesting species that lays eggs on land, unlike the live-bearing species that we've just been looking at, and mostly eats, we think, sirens and amphiumas, so giant aquatic salamanders. And this is one of my major motivations to work on the Savannah River site, because mud snakes are really cool and there's a lot we don't know about them. So they made up about 5% of our captures. It has another species in the same genus, Varantia erythrogramma, the rainbow snake, these making up less than 1% of all captures, and these eat mostly eels. And then finally, cottonmouths. Cottonmouths are really abundant in these systems, but they don't actually forage in a way that's very similar to the other 6 species. So they're not active foragers, they're more passive foragers, meaning they sit in one place and wait for their prey. And so they're not as amenable to trapping. So even though they made up 10% of our captures, most of those were not captures using minnow traps. Most of them were captured outside of minnow traps, but they are really abundant there. And so, and occasionally we would capture other species as well. So 2 other species of water snakes here. Very rarely we would catch a rough green snake or an eastern king snake that would enter one of our traps if it was placed in shallow water. But this really is a method that's best for detecting fully aquatic species. Okay, so whenever we would capture snakes, we would bring them back to the laboratory, and the most important thing that we would do would be to mark them. So we do this in one of 2 ways, either by clipping off the margins of their ventral scales in a pattern that we could read later. Sometimes we would also use a medical cautery unit to heat brand those snakes, and you can see kind of a schematic that shows the system that we would use on the left there. So they would all get individual marks for mark recapture, and we would hope to recapture some of them and be able to say something about the second question, how many are there? And that was kind of a major cornerstone of the dissertations of 2 of the graduate students that I was lucky enough to work with when I was an undergraduate there, J.D. Wilson, who I've mentioned before, and Chris Winnie. And these guys were doing their dissertations on communities and populations of aquatic snakes, mostly at Ellington Bay, and they were embarking on this major capture, mark, recapture effort, including an effort to estimate the imperfectness of the detection process. So how often are snakes present, but you actually don't find them, or how often are individuals present in the population, but you don't capture them. And so the way they did this was it's really a lot of effort, very labor intensive process. So they would set 465 minnow traps 2 meters apart around the perimeter of Ellington Bay. They would do this for 6 to 10 nights over a period of 8 months. And they generated over 3,000 captures of over 1,000 individual snakes just within Ellington Bay alone. So this is just the focal bay, okay? And you can see that they documented pretty serious seasonal variation in capture rate. So they captured a lot more, particularly black swamp snakes in certain months than others, you know, maximum 400 captures in a month, minimum would have been maybe 20 or something like that. But overall, they really were able to generate some nice estimates of population size for the 2 most common species, banded water snakes and black swamp snakes. And their population size estimates were similar, between 200 and 300 individuals of each species but nearly doubling in the fall following reproduction of these two live-bearing species, which give birth to their litters in the water. So this is really cool because it allowed them to generate a really robust estimate of population density within Ellington Bay, and that was 171 snakes per hectare, or if you take their biomass into account, 7.7 kilograms of snake per hectare. So that's really quite a lot of snake biomass in a very small area. And in addition, they documented a couple of other really interesting things about the ecology of these snakes. One is kind of an element of the actual sampling process. You can see that there's this category called recent recaptures. So these are snakes that had just been recaptured maybe the day before or 2 days before, certainly within the same span of those 6 to 10 consecutive nights. And animal population ecologists call this phenomenon trap happiness. So they were able to show that particularly banded water snakes were up to 5 times more likely to be recaptured after the first time they had been initially captured. And you know, we think that that probably has to do with the fact that they like to go in those traps because they know that they often get a free meal in there, and they do. You might think, well, they would avoid them because they get kind of harassed afterwards. But if they go in them often enough, probably most of the time they're quickly released because JD and Chris and I would recognize them and read their marks and know that we had just caught that one yesterday, maybe even in the exact same trap. So we would release them. And so they would have positive experiences with the traps maybe more often than negative experiences, which is quite interesting, and I think something to keep in mind for anyone doing this kind of work. The second super interesting thing is this interaction that they documented in black swamp snakes between what's called temporary emigration, or sort of temporary unavailability for capture. So that's that imperfect detection thing that we were talking about. Interaction between that and sex. So they found that about 20% of male black swamp snakes and 36% of female black swamp snakes were unavailable for capture in any given month, meaning that those snakes were often captured and then not captured and then captured again, often fairly reliably. And so they thought that this represented individuals leaving the capture area. So remember, I sort of said that the traps are set around the perimeter where it's the appropriate depth to set them. And this area kind of moves as the hydroperiod changes, but you don't want to set these traps in an area with 1 meter deep water because the snakes inside them will drown. You really want to set them in shallow water where there's enough headspace of air that the snakes and the other animals that you catch can breathe air. And so they thought that the black swamp snakes likely had smaller home ranges than the banded water snakes, and so sometimes they would move out of the trapping area and back into it. They may have also been doing this for another reason, which I'll come back to when we talk about drought, and that's particularly important in terms of the interaction with sex. So more females were likely to maybe leave the area and return. So these are insights into the behavioral and population ecology of these animals that would be very, very difficult to obtain by any other method. And I want to emphasize that, you know, this slide represents just years of field work. And these guys and I spent so much time doing this whenever they were doing it. And that, it's super labor intensive, and in general for snakes, the recapture rates are pretty low. So this is a unique opportunity where the recapture rates were actually high enough for them to get some good estimates. But as many of you know, if you've tried to work on especially terrestrial snakes before, there's really no analogous method that works as well as these minnow traps for capturing and recapturing terrestrial snakes. And so a similar study on even a common terrestrial species like a racer or a rat snake probably wouldn't turn out nearly as well. So it's really impressive work. And so this is like the foundation. I mean, this was ongoing at the time that I was there, but this was the conceptual foundation that my work is built on. So my contribution to this project, in terms of, in the spirit of full transparency, I was, 18 years old when I was at the Savannah River Ecology Lab, and I turned up day one, and I think I just told JD and Chris, well, I really just want to catch as many snakes as I possibly can, because that's what motivated me at the time, and it's what still motivates me a lot of the time. And so JD suggested, well, why don't you look into this occupancy modeling stuff? Because this would be a good method to look at all the different wetlands around the Savannah River site pretty easily and maybe compare them in terms of their hydroperiod and other things, and learn about which snakes are where, and a little bit maybe about how many there are, but mostly trying to get at that first question of which snakes are where. So let me just walk you through occupancy modeling in a conceptual way quite quickly. So most biologists for most of history have collected what we think of as presence-only data. It's pretty intuitive. Every time you see a particular snake, you would write down you saw that species, the location, the date and time, maybe how many, right? So many people's field notes sort of look like this. And it wasn't really until a little bit after the turn of the century that some statisticians started to think instead in terms of what they call presence-absence data. So these are the same data on ribbon snakes, and you can see the same couple of sites are in there, same couple of dates, but there's actually a third site, Site C, that was visited 3 times, and ribbon snakes were never detected there. So that's quite important because that information is lost under the presence-only scheme of data collection, which is, as I said, sort of the default. It's the one that most people probably still use, and it's the one that maybe makes the most intuitive sense to people. But these absence data are really important. So, you know, if I tried to tell you that Site B was not occupied by ribbon snakes on March 10th, but that it was on the 9th and the 11th, would you believe me? Probably not, right? Because you can see that individuals were captured the day before and the day after, and we know that snakes are very secretive, and so maybe, you know, the weather wasn't as good that day or something like that, and so none were seen. If I tried to convince you that Site C was unoccupied by ribbon snakes, you might believe me, you might not. But the beautiful thing about occupancy modeling is that it kind of ties these two things together, these two things being occupancy, or the probability that the species is actually there or not, and detection, which is the probability that you saw it, given that it was there. And these are two different things that are really important concepts in population ecology. Another way of sort of thinking about it is that in the presence-only data framework, you really can't tell if a species is changing its abundance because of imperfect, or because of something in the environment, like maybe it's going extinct, or it's colonized a new site, or it's doubled in abundance. You really can't tell that from something about imperfect detection or unequal sampling effort, because you don't have any of those zeros. You don't have any of that absence data. And a shorter way of saying this, maybe more familiar, is that absence of evidence is not evidence of absence, right? This is something we're all thinking a lot about right now with the COVID pandemic, false positives and false negatives and things of that nature. And so you really do need to do a lot of data collection that doesn't on the surface seem very useful, and collect a lot of zeros or absence data in order to make your presence data more useful. So presence absence data are good because they can account for imperfect detection, they account for sampling effort, and they can help you detect changes maybe in at least relative abundance. It's not quite as useful for that as mark recapture, because you're not actually able to say anything about the number of individuals that are there, but it's probably better than nothing, particularly for rare species that have really low recapture rates. So let me show you what we did with occupancy modeling. So this is you can conceptually think of this, if you want, as similar to mark recapture, but really at the species level. And so we, you have to have multiple sites when you do this. So we picked those 20 wetlands that I described earlier, and we set 30 to 90 minnow traps in each one. We trapped each one for 5 consecutive nights. And this was possible for me to do in a summer because the trade-off between the number of sites you can include and the number of surveys you have to do at each site is not as intense as it is for mark recapture. I mean, we got way fewer recaptures. I think I have the number right here. Yeah, 319 captures of 230 individual snakes. So relatively few recaptures, right? Fewer than 100, whereas JD and Chris had over almost 2,000 recaptures from their Ellington Bay data. So we wouldn't have been able to say very much really about the abundance of snakes in these wetlands. But what we can say is which wetlands were occupied and which ones were not. And if they were not occupied, we can say what's the probability that they were really unoccupied versus the probability that we just missed them, right? That the detection there was imperfect. So instead of estimating abundance and density, you estimate this thing you call occupancy. And you can see for the 7 species here on the right, we get estimates of both occupancy in the light gray bars and detection probability in the black bars. And it varies a lot among species. So some species like banded water snakes, they're very detectable, and they're also very widespread. So they have a very high occupancy probability, right? Almost every wetland is occupied by them. And if they're there, you're likely to catch them. Species like mud snakes are also fairly widespread, it seems, but their detection probability is much lower. you're less likely to catch them. Maybe that's because there's fewer of them. Maybe that's because their behavior is different. They can leave the wetlands a lot more easily, right? And maybe move from wetland to wetland. So that's an important difference. Then you have species like black swamp snakes. So their detection probability is relatively high when they're present, but they're not very widespread, right? They like certain conditions. They're very rare or local. So their occupancy is low, but their detection is high. And then you have species like rainbow snakes where both occupancy and detection are low, right? This is a very, very probably legitimately rare snake. It's only found in certain habitats and it's very secretive, so it's difficult to detect it when it's there. And these are the hardest cases, right? So hopefully this gives you some idea of what occupancy modeling can do for you and how we were able to kind of apply this to these secretive snake species in a relatively short time frame and generate some really useful information. Another thing you can learn from occupancy modeling is how many visits you would have to make to a particular unsurveyed site to say with some confidence that the species is actually not found there, right? And so here's what that looks like. Some cases it's as low as 4 or 5 visits. In other cases, you know, for rainbow snakes, you're looking at 30 visits, right? So in our case, a visit is 30 traps set for one night. And cottonmouths is even worse. So I mentioned earlier that we don't think that our method actually works particularly well for detecting cottonmouths because they're ambush foragers. But even 30 visits is a substantial amount of effort, right? I guarantee you that most biological survey companies, for instance, when they're surveying a piece of property before it gets developed, they're not making 30 visits to that property, right? They might be making 5 or 6. So I think this is really important from a conservation perspective. Now, I don't want to run out of time, so I think what I'd like to do is just skip ahead a little bit here. Actually, let's just take a look at the next 2 slides, and then I'll open it up for questions. So after I left the lab, I continued to stay involved with JD and Chris, and so several years later, you can see that there have been some periodic droughts at the Savannah River site, and shortly after I left, there was sort of the worst drought in well over a century. So there was very little rain at all, and the wetlands hardly filled up. A lot of them were totally dry. And so Phil Vogrens, which he was one of JD's master's students at the University of Arkansas, he basically had the idea to repeat the sampling that I had done in 2006, but following this really severe drought. And we were interested in looking at changes in the occupancy of these different snake species to understand better how did they respond to drought in the landscape. And so what we found, I'm just showing 2 species here because these are, I think, the most interesting contrast. What we found is that some species like green water snakes, their populations really crashed. A lot of the wetlands that were occupied by green water snakes before the drought were not occupied by them after the drought. And we can exclude the possibility that they were still present there but undetected because we estimated and accounted for that imperfect detection. And this kind of makes sense. I mean, we think of this green water snake as really being a species that relies heavily on permanent water. So probably what's going on is that there's a strong metapopulation dynamic in the different wetlands on the Savannah River site where very few sites, maybe spring-fed sites or sites that are close to the river floodplain, are the sources of the green water snakes that would colonize the other more ephemeral wetlands during a wet year, but go extinct there during dry years. In contrast, these teeny tiny little black swamp snakes, which you would think would be super vulnerable to drought, their occupancy really didn't change at all throughout the drought. And that's amazing. And this, I think, is one of the things that makes this one of the most interesting snakes out there. Black swamp snakes are like a lot of amphibians in that they have the ability to actually estivate in the mud at the bottom of a dried wetland, sometimes for many years. And these, the ecology and physiology of black swamp snakes was the major subject of Chris Winnie's dissertation work. So he was able to show, for instance, that even though the populations could persist through droughts, the body size changed a lot. So prior to the drought, maybe 30, I think about 1/3 it shows of all the black swamp snakes captured in Ellington Bay were greater than 300 millimeters snout vent length, and 2/3 were lesser. After the drought, 98.5% of them were under 300 millimeters snout vent length. So there's a strong selection to be small if you're going to survive a drought. And that has kind of profound impacts, I think, on their physiology and also on their population ecology, because a lot of times they can reproduce right after the drought is over, which is really amazing. Snakes are just awesome like that. Okay, final thing I'll show you here is a result from the occupancy model that I did, where you can actually graph and look at how something like detection or occupancy changes across an environmental gradient. So what you can see here is the less permanent, more ephemeral Carolina Bay type wetlands on the left with very few fish, fewer sirens, and on the right, the more permanent, more fishy wetlands. And you can see how something like a mud snake is more likely to be captured in a more permanent wetland than a less permanent wetland, but that they are found sort of across the spectrum, whereas something like black swamp snakes or banded water snakes are way more detectable in these less permanent systems where they've really, black swamp snake in particular has become a specialist of these ephemeral systems, and it can persist there even through long droughts. And really in the most permanent wetlands, they don't do particularly well at all. Maybe they even fall prey to large fishes or something like that. So, all right, I don't want to run out of time, so I'm just going to jump ahead and say thanks to everyone who helped me collect and analyze these data, particularly the NSF REU program. And I would be super happy to take any questions, or if there are no questions, I can tell you a little bit more about the third bit, which is what snakes eat.
Thanks, Andrew. I can help. There are a couple questions about what we've just talked about, but we'll see how that goes. And then, yeah, we can always hear some more. So we had a question from Scott who asked, if snakes migrate substantial distances in times of drought, is it possible that rather than being truly extirpated in formerly wet areas, could they just be finding larger bodies of water?
Yeah, excellent question. So like many things in biology, the answer is it depends. But JD and Chris and I thought about that. They thought about it hard. And I think in the case, in the best known case, which is Ellington Bay, there are very, very few records of snakes migrating out of Ellington Bay during the drought. So the closest other bay, ironically, is a bay called Dry Bay. It's a bay that doesn't dry out quite as often as Ellington Bay, but I suppose it was named first or something like that. And there are a few records of water snakes moving from Ellington Bay to Dry Bay, but that's a pretty long distance. They have to cross a pretty big road. And during the study period when JD and Chris were doing their most intensive sampling, there were people, someone else, a master's student by the name of Tom Luring, was doing similar trapping at Dry Bay. And they never documented any inter-wetland movement. So what really seems to be the strategy, at least for banded water snakes, is that if their wetland dries up, most of them are going to die. Black swamp snakes, of course, they can do the estivation behavior like we talked about. So it's really, I think their strategies for persistence really vary depending on both the species and where they're found. If there is a permanent water body nearby, like for some of the wetlands that are close to the floodplain, probably they do leave, particularly the ones like mud snakes and rainbow snakes and cottonmouths that are good at dispersing over land. They probably do leave, but something like a black swamp snake basically can't disperse over land. Its ability to colonize new wetlands is limited really to times of the year when there's a lot of flooding, and maybe it's moved around that way because their rates of evaporative water loss are really high, so they dry out if they try to disperse over land. Super good question.
So just to follow up on that, I was curious, and then there's another couple questions, but do you think they use crayfish burrows to estivate? Is there any information about that?
So a lot of small aquatic nature scenes do use crayfish burrows, crayfish snakes, for instance, but also kirtland snakes in the Midwest and lime snakes in the Great Plains. I would not be surprised if certainly glossy crayfish snakes were using crayfish burrows in some sites in the Southeast, particularly higher, drier sites. Ellington Bay is really unique in that it actually doesn't have any crayfish either. So it's a very, very simple prey community at Ellington Bay, and so there's no crayfish burrows there for them to use, and yet they seem to do super well though. So sometimes.
Cool, thanks. So we had a question from Sabine. She said you specified the impact of drought on the snake population and the snakes itself. What about the impact of hurricanes?
Ooh, awesome question. I've not worked on this myself, but I know some people who have. So Margaret Gunsberger did her, I think, dissertation work on the impact of hurricanes on amphibian communities in isolated wetlands in the Florida Panhandle. She may have looked at snakes as well. I'd have to check. And in general, I think what she found is that largely there was a lot of evidence for positive impacts of hurricanes on amphibian and maybe by extension reptile populations. You know, sure, some of them are going to be killed in the storm surge, or you know, maybe a tree falls on them or something like that. But in general, it's good for their dispersal, gets a lot of nutrients moving, and probably causes a big boom of resources right afterwards. And so it seems clear to me that for aquatic snakes, the impacts are normally quite positive. I'm also thinking of a study that was done by Al Savitsky and J.D. Klopfer in the Virginia Beach area, where they actually showed that tree falls caused by hurricanes in deciduous woodlands were really important basking sites for timber rattlesnakes in those areas. So before hurricanes, when there were not a lot of gaps in the canopy, all the rattlesnakes were like clustered around this one sunny spot. And then after the hurricane, they could disperse and move out and bask in different areas. And so it was quite positive for them as well.
Cool. So here's a follow-up question from Dylan at Archbold. He's related to the hurricane question. Did you ever, is it possible to get large rains that flood so much that fish do get introduced into non-fish wetlands like eBay?
It is. And I think, you know, I'd have to go back to the Siegel papers to see what the situation was like at eBay in the 1980s. I think there is some evidence that, if not fish, then at least there used to be crayfish there, if I'm not mistaken. Maybe you know. But I think what ultimately happens in a lot of those really ephemeral wetlands is that if there's a very rainy time, then it's often followed by quite a dry time. And so there's a local extinction of those, fish or crayfish or whatever it is that get introduced. In Florida, it's quite different. And what I'm hoping to do, what I meant to talk about more is, you know, in southern Florida, a lot of our wetlands are connected right now because we're having tons of rain right now, and we kind of have no rain for 6 months every year. And so I'm really interested in how these same processes play out in communities that contain a lot of the same species, but in an environment where the hydroperiods are a lot more dynamic within the year and maybe also more predictable, although it seems to be, I'm still learning a lot about southern Florida. So if anyone has ideas, that's something I'd love to come do at Archbold, because I think you guys have maybe more wetlands that would be amenable to something like this than even the area where I live.
Yeah, I agree. It'd be super interesting to compare what's going on, because it is dynamic is the word.
Yes.
So we had a question about the diets and how you really go about knowing the diet. So is it correlative? So you know, ephemeral ponds have no fish, so snakes there don't eat fish, or is there real experimental observational, you know, information to back up statements about what they actually eat?
Yeah, great question. Thanks, Austin. And also, hi, hope you're doing okay. So it's kind of both. And, you know, snakes are hard because they don't eat that often, but I'll just show some data here that I meant to show before on the prey availability at the different types of wetlands. So we measured this by counting all of the prey that we caught in our minnow traps. So aquatic salamanders, adult and larval frogs, fishes, crayfish, and then some other invertebrates as well, things like leeches. There's a category in here for other snakes, I think, because that's something that at least cottonmouths sometimes eat. And then we looked also in the stomachs of these snakes and compared prey availability with what they were actually eating. Major criticism being that, of course, they do like to go into the traps to eat things there. But because we were exclusively in our experiment making comparisons of trapped snakes to other trapped snakes, we argued that that bias maybe wasn't so bad. But a lot of snakes eat rarely. So I'll also show you this really quickly. So this is data from sort of my survey that I've been doing of studies that report how often the stomachs of snakes are empty. So I've got almost 300 studies in here, and the median percent empty is almost 75%. So there are a lot of missing data here, a lot of absences. And that, for my master's and my graduate work, really got me thinking about other ways to measure snake diet. And so Betsy, you mentioned stable isotopes as a promising one, and it is, but it also has some weird drawbacks that I can talk about if anyone's interested. And so more recently, I've been thinking about combinations of kind of traditional gut content surveys together with fecal eDNA surveys. So either swabbing the cloaca or getting a fecal sample, which most snakes, when you catch them, are happy to give you, and then sequencing the DNA of the prey that are in there, maybe also with stable isotopes, but they're also very labor intensive, and sometimes they don't actually answer the question that you think they're answering. maybe I can do another talk about that sometime because it's interesting.
Right. So that would be great.
Sure.
So we had a question about just what was that beautiful snake on your questions page?
Yeah, so this is a juvenile mud snake that I photographed. This was probably my favorite snake that I caught for this entire project because they're just, I mean, mud snakes are beautiful, period, and the babies are the most beautiful. And this is kind of faded out, so you really can't even appreciate the intensity of the bright colors. But man, they are so cool, and we just don't know that much about them. We really don't even know what the babies eat. They're pretty hard to keep in captivity. A couple of people have managed to do it, but this is an open area. I think ecology of juvenile mud snakes is something I'd love to do more.
Yeah, detectability doesn't get any easier as they get smaller.
No, that's certainly true.
Okay, we have another question if you're up for it from
Sure.
Kyle. Let's see. Okay, he's asking about the gap in the range of Neurodia floridana between South Carolina and Florida. So, and whether it could be driven by land use change, resistance to disperse overland during drought, just disjunct the gaps in artifact, you know, what's going on there?
Awesome question. I think Tristan Tramer's in the audience. He's probably thought about this more than I have. So Tristan, if you're still here, feel free to chime in. I think it could be a combination of those things, but to be honest, I've been living outside the Southeast for long enough that I don't know. I don't know if anyone knows. Are you here, Tristan? Sorry to call you out if you are.
Can you hear me?
Yes.
Okay. Yeah, that's a tough one, and I don't know if anybody really knows the answer. You know, it's one of the big key questions about Floridana in Whitfield's water snake book. But hopefully, if I get some more grant money for my thesis work, we'll be able to do some low coverage genomes in both parts of the range to see if there's actually a genetic signature of like historic break, or if it's more of a recent occurrence.
Yeah, I mean, Georgia is not exactly undersampled, I would say. Although, you know, you could also argue that sort of everywhere is undersampled for snakes, particularly in contrast to the Savannah River site. I mean, if you look at the total number of black swamp snake records from the Savannah River site compared to anywhere else, maybe outside of southern Florida, you know, Kings Prairie, maybe something like that, it just dwarfs it, right? Because you know, that was preserved, I think, in a way that's very different from most parts of the Southeast. Excellent question.
And I just realized I missed an early question from Doug on the whole Carolina Bay mystery. Because they have that elliptical shape and they're sort of consistently aligned from northwest to southeast. So he was wondering why that was.
Yeah, again, I'd probably refer you to a geomorphologist if there are any in the audience. You know, I think that's one of the big pieces of evidence that led people to suggest maybe it was a meteor shower, but I'm assuming that if it is not a meteor shower and in fact is kind of wind and water working together on sort of pooled water that was left behind during the receding, you know, during the time when the ocean was receding away from the former coastline, may have something to do with the consistent direction of the prevailing wind. I mean, it's kind of the same question of like, well, why do thunderstorms always come in, you know, off the water, right? It's like maybe those forces over time always align, but they are like almost eerily aligned, right? So I think a geomorphologist would probably have a much more intelligent answer than that. That's all.
Yeah, so when I was at the lab, I remember, because I did my postdoc at SRAL, Andrew had alluded to us almost overlapping, sort of.
Yeah, I think we might have missed each other by about a month or something.
Yeah. But I certainly remember all this snake trapping going on. But I think I remember seeing our cartoon that was floating around the lab, which showed a dinosaur golfing as a potential explanation for all these little divots that were all oriented the same way or something.
That's awesome.
Anyway.
That's my new leading theory.
Yeah. Okay, I'm not seeing other open questions. Am I missing any? I'm looking at the chat.
Yeah, take a look at the chat, Betsy.
Um,
Scroll up a little bit and you have a couple from Susan. Susan asked, you mentioned about having more slides. Is there a place that they can view the entire lecture?
Sure, I'd be willing to post it on, I don't know, do you guys have a place to host it? I guess I could post it on Figshare or ResearchGate or something like that and make it open.
we have, I don't know, yeah, like the static slides. Maybe we can figure that out. Stay tuned. We'll figure something out there.
Yeah, sorry, I probably
right.
tried to put a little bit more in here than I should have.
We'll put it in our follow-up email for sure, Susan.
Okay, so I do see a question from Ellen Rivero here at Archbold. He's asking, did you notice similar recapture rates with crayfish? I'm not sure I understand. Similar
With
recapture rates with crayfish snakes.
Yeah, crayfish snakes are kind of an enigma. So I think that crayfish snakes are even more local than black swamp snakes, at least at the Savannah River site. Obviously, they need crayfish, and that's kind of one of the take-home points from the diet study. Is that I'll just tell you in a nutshell. What we found in the diet study is that almost all of these snake species were found even in the wetlands that had the fewest prey types. In particular, wetlands like Ellington Bay that don't have any fish, they don't have any crayfish, they don't have any sirens or amphiumas. So there's very little for snakes to eat there, except for the fact that there are tons and tons and tons of larval amphibians, especially larval frogs, randid frogs, some hylids also, and embistomatid salamanders. And what we found is that basically every snake in Ellington Bay was eating embistomatid salamanders, and that's everything from, banded and green water snakes, black swamp snakes, mud snakes, rainbow snakes, and cottonmouths, all living together and eating these super abundant embistomatids. And you'll notice I didn't say black swamp snakes there, so, glossy crayfish snakes. So no crayfish snakes are found there, and that's because there are no crayfish, I think. So they seem to be the only species in this community that is, or the species in this community with the most inflexible diet, right? They really are specialized for crayfish. So in the places where we found them, they were, I wouldn't say abundant. I think there are some wetlands on the Savannah River site where they are abundant closer to the floodplain of the Savannah River itself, but they're hard to access. So we didn't trap a lot of them. There were a few small wetlands where we had decent recapture rates for glossy crayfish snakes, but I would like to do a study focusing specifically on them. Unfortunately, we don't have them in southern Florida, but we do have Allen's crayfish snakes, which are ecologically quite similar. And I think that, yeah, they're kind of chronically ignored. Everyone's like, ooh, mud snakes. And don't get me wrong, I love mud snakes, but there's also a ton we don't know about crayfish snakes.
All right, so I just had another question come in. We can keep you going all afternoon.
I know.
So Sabine is asking, do you have any snake projects in mind at FGCU, and would you accept volunteers?
Yeah, so I would accept volunteers. If I had any snake projects going on currently, I meant to have some ongoing by now, and obviously the pandemic has kind of slowed that down. So when I do have them rolling, hopefully by this fall, I'm hoping to have everything in place to actually start setting some traps and doing some market capture and some occupancy modeling in our campus wetlands. At that point, yeah, I would be happy to accept volunteers. I have some students that I'm beginning to get to know and work with. That process has also been made more difficult by the pandemic, but yeah, that's the plan. So shoot me a private message, an email, or however you want to connect. I'm happy to talk to you about that if you're local.
Good. I hope you can get that off the ground sooner rather than later.
Me too. And I'd love to, to bring some traps to Archbold and do some similar studies up there, maybe over a summer or something.
That would be great.
Yeah.
That would be awesome.
Cool.
And then I guess maybe last question here. Kevin on Facebook wants to know, how did you handle those cottonmouths that were The ones that you did catch.
Yeah, so you know, very, very carefully and safely, always with snake tongs and a snake hook, never putting our hands on them. The one that I showed the picture of that had gotten tangled in the entrance to that minnow trap, that was the single most challenging snake handling experience of my life. So I haven't really worked a lot on venomous snakes except for the cottonmouths in this system. Ever since then, I did my master's on hognose snakes. I did my PhD work on mostly lizards, but a little bit on garter snakes. And so getting that snake unstuck was something that JD and I, my mom and my brother were visiting at the time, worked on for probably about an hour, just sort of slowly easing it backwards out of the entrance to the trap without freeing it too quickly, right? And then kind of switching over to the snake tongs right at the point when we thought, okay, it's just about loose enough that we could pull it free with the snake tongs without hurting it. So yeah, we had very, very strict safety protocols in place at the Savannah River Lab for handling cottonmouths, and also copperheads and timber rattlesnakes, which we would occasionally catch on the roads or something like that.
Yeah. Yeah, you got to be prepared to handle whatever goes in those traps.
Yep. You know, in a sense, it was kind of a relief that we weren't catching most of the cottonmouths in those systems, because it would have made the work a lot harder to do, because, you know, opening a minnow trap and scooping out a couple of black swamp snakes or banded water snakes and putting them in a bag and moving to the next trap doesn't take very much time. But every time you find a cottonmouth, it takes a lot more time to bag it up. And so we did, Evan Esky was a high school student in the lab at the time, who's now with the Eco Health Alliance in New York City. And he did a really good study on cottonmouth foraging and habitat use when we kind of went out to Ellington Bay at night and walked around and captured cottonmouths and marked where they were and things like that. And that was fun, but yeah, it's a lot of work to safely do research on venomous snakes.
Yeah, whole other level.
Indeed.
Complexity going on there.
Yes,
all right, well, I see that it's about a little after 4:30, so we should probably wrap up. But I just want to thank Andrew again and everybody who joined in. And Laura, I don't know if there's anything else you want to tell people about or not, but yay, Andrew, thank you so much. It was so fun hearing about SREL again.
My great pleasure. It was so fun for me to put this together and delve back into some of these things that I haven't thought about for a while, but that I'm hoping to rekindle here in Florida. So thanks for having me. And thanks everyone for coming.
Yes, thank you so much, Dr. Durso, and thanks to all of you for participating today. We do not have a Thursday seminar next week, but you can tune in on Tuesday morning at 9:30 when Dustin Angel takes us to Lake Annie for our last June virtual field trip. Then our Thursdays resume on the following week on July 9th as Scott Ward presents Endemic Plants of the Lake Wales Ridge. See you soon.
Bye.